Hello everyone, welcome to my presentation. My name is Mujtaba and I'm from Louvain, biomechanics section, actually Department of Mechanical Engineering under supervision of Professor Lisbeth Gieris. As you may have noticed, the title of the presentation is a little bit changed. That's why originally I was supposed to talk about mathematical models, detailed mathematical models for tissue growth and cell proliferation. But then after discussing that with my supervisor, we came up with the conclusion that that's too much. So I changed it to a more general presentation about computational modeling in tissue engineering. And then, for more specific examples and use cases for tissue growth as well as degradable materials, which is actually my main line of PhD research. So to begin with, I will have a general overview of computational tissue engineering, why it's actually necessary in the 2021. It's actually taking advantage of development of mathematics over a couple of centuries, as well as taking advantage of recent computing powers, available computer powers and advancements in computer architectures to be able to optimize tissue engineering products as well as, let's say, processes, and at the same time assessing the influence of different parameters on this stuff. So that's why actually this is an important field, although this is tissue engineering is more dominant by experimentalists, but the computational works can be beneficial in this perspective. But what is really important to discuss is when we talk about modeling, we should pay attention that there are a wide range of different types of models. So models can be data-driven or based on first principles, or in another classification, they can be deterministic or stochastic, or they can be mechanistic or phenomenological. And for example, for just the last class, classification, I can say that they differ a lot. So when you talk about mechanistic models, you mean that they require more data, more assumptions, and they can handle more complexity in comparison to models that are based just on phenomena. But at the same time, they can cover more scope, let's say. So they are more diverse. What I will discuss in this presentation will be more focused on deterministic models and mechanistic models. This is an example, actually, this is not something that I have done, but this is an example that states the importance of multi-scale modeling. This is a trend in tissue engineering you have definitely heard about. But it means that you study, you investigate phenomena on different scales, in different length scales or time scales. For example, this is a stochastic model of tissue formation in rats without BMP, and then at the same time in another scale, you have a deterministic model of cell proliferation. So you have models in tissue scale, organ scale, cell scale, intracellular scales. This is, for example, for the cell scales that you can solve stuff continuously and investigate cell proliferation for bone healing applications. Or at the same time, this is also kind of model, kind of results that you will see a lot in this field that is related to stochastic, semi-mechanistic stochastic models for bone and cartilage formation. So different colors for bone and cartilage separately. This is actually done by Eduardo, a researcher in our group, has been recently published also. But the general workflow actually is, for any kind of model, it is converting underlying sciences to mathematical models, and then, and then mathematical models to computational models. And when we talk about underlying sciences, it means like the different principal laws, like laws of biology, laws coming from physics or chemistry. And they can be converted to mathematical forms that are usually equations that correlating different parameters or different phenomena from these basic principles. And then for computational models, it is usually the stage that you use computing power and numerical techniques or analytical equations to solve the mathematical models. And this is actually the workflow that is very typical in these kind of projects. So the first use case, as I said, this is my main line of research and for doing my PhD, is a use case for biodegradable materials. What happens to biodegradable materials inside a body? Talking about biodegradable materials and metals, actually, we are referring to zinc, magnesium, and iron, and they gradually disappear and get absorbed by the body. They have great mechanical properties, but also positive contribution to the human body metabolism. But then the problem is the release profile should be controlled and tuned for different applications. And this is where computational modeling can come into play and help to tune it without performing in vitro and vivo experiments. So in this case, if we want to talk about a workflow, the underlying science would be the chemistry of biodegradation and material interaction. And when we talk about mathematical models, it means differential equations, or more specifically, partial differential equations correlating different chemical components interactions to each other. And computational models is actually solving the equations that we have derived during construction of the mathematical models and solve them using numerical methods like the famous finite element method you may have heard about. And then, qualitatively, these are kind of results that we can get, for example, for orthopedic implants. That is actually how, the video, I think, it's a bit heavy to be rendered. But this is, for example, on the left, you can see the release of material and the degradation of the, let's say, in this case, implant, and on the left, just the surface of the implant being degraded over time. So we can change parameters, investigate different things, let's say, in silico, in a virtual world, and then see what happens to the degradation profile. And this is for open porous scaffolds for tissue engineering, that what happens to those kind of scaffolds, and allowing us to really optimize the structure to improve the rate of tissue growth when it's combined with tissue growth models. And this is for another project, actually, for a jawbone implants, for the degradable ones. And it was supposed to be coupled with other mechanical models to investigate what happens to the mechanical strengths of these materials over time when they start to degrade. And also this one is a simple cuboid that was initially used for degradation, for validation of the models. Sorry, the video doesn't work, so I don't want to put burden on this computer. And then for validation, it's not usually always qualitative. There are lots of quantitative output that you can get and then compare using experiments by in vitro and vivo experiments to validate the models. And the second use case is for tissue growth. That was actually the original presentation. So it's actually about cell growth and open porous scaffolds, a different effect of different geometrical and structural, let's say, characteristics and properties. And one solution is computational modeling of tissue growth. In this case, this is actually tissue growth on a scaffold inside a perfusion bioreactor. And then I will briefly discuss it, that this is categorized in a class of moving interface problem, that we track the moving of the interface between the growth, the tissue, and the void space. And by that time, this is a special class of mathematical problems. We can actually use those kind of advancements in mathematical formulation to track tissue growth in tissue engineering. So in this case, the underlying science is the biology of cell proliferation. A mathematical equation is, again, differential equations describing the movement of this free boundary. A computational model is numerical methods to solve those equations. So for capturing the interface, actually, when I was talking about moving interface, it was actually the tissue and the void, the interface between them. So it's crucial to capture this. So this is the interface, and then we formulate a sort of equation that correlates the speed, the velocity at which the interface moves. And by doing that, I'm just trying to determine different affecting parameters to, for example, coming from biology or from physics to this, we can model the tissue growth. So this is an example of one of the affecting parameters, that is actually the curvature of the tissue at the moment that is growing. So the higher curvature is, faster the tissue grows. As you can see here, that when the curvature is higher, we will consider more velocity, actually, to be said in a simplest way. And for example, here in the 2D form, cells sitting around, you can see that the simulation will be something like this. And then in 3D, using same, it shows the same behavior, let's say. And it can be extended to compute more complex scaffold shapes like gyroids or even more complex types. And this is actually how it works in silico models for gyroid-like scaffolds. And again, by validation, this is actually we had a discussion with a friend here that the validation is the most difficult part in this kind of research, that you need to validate the models because it's easy to reproduce everything inside a computer, but in the end you need to validate it. And in this case, validation can be done qualitatively, like comparing the tissue growth profiles to imaging, to the images that you will get from tissue growth and cell proliferation, but at the same time it can be also quantitative, comparing their tissue growth rate for different shapes and trying to really finding the similarity to the trends in quantitative data. So the conclusion, I talked it's very short presentation about the role of computational model in tissue engineering, and then a use case one for computational modeling of tissue growth, of material degradation, and the use case two for the semi-quantitative computational model to investigate tissue growth. And once fully validated, these models can save lots of time and money, and can be replaced the expensive actual experiments. So thank you so much for your attention.